Hi, everyone. It is my pleasure to introduce Dr. Corrada for our MAPP round today. I just wanted to remind all the attendees to write any questions that they may have throughout the presentation in the Q&A, and we will be discussing them. We will be answering all of those questions at the end of the lecture today. We'll have kind of like the 10 last minutes for to answer questions. So let me introduce Dr. María Corrada. She's a professor in the Department of Neurology and the Department of Epidemiology and Biostatistics at the University of California, Irvine. She has an undergraduate degree in biology from the University of Puerto Rico and completed a master's in biostatistics and a doctorate in psychiatric epidemiology at the Johns Hopkins School of Public Health in Baltimore. Dr. Corrada is multiple PI and co-founder of the 90+ study, a longitudinal epidemiological study of aging and dementia of more than 2,000 people aged 90 years and older in Southern California. She's also multiple PI and co-founder of Life After 90, a life course study of cognitive impairment in a multi-ethnic cohort of over 1,000 people aged 90 and older in Northern California. More recently, Dr. Corrada has been in collaborations exploring the health burden of Alzheimer's disease and related dementias on American Indians and Alaskan Native elders. Her research interests include population-based longitudinal studies, the epidemiology of dementia, identifying factors that correlate with the presence of brain pathologies, dementia, and cognition in ethnically and racially diverse populations, and in particular, understanding the cognitive and physical health of the oldest whole population. So it's our pleasure having you today. So Dr. Corrada, the floor is yours.
Well, thank you, Marta, for the introduction. And I'm very happy to be here. I have to say that I have always admired the work of Dr. Quiroz. So when she invited me, it was an honor and had to immediately say yes to being there. So thank you, Dr. Quiroz, for the invitation. And so I guess I have nothing to disclose. And I kind of wanted to talk about like the two cohorts that were mentioned in the introduction. And I wanted to basically discuss, why do we care about the oldest old? Why do we need to study the oldest old? And most research in Alzheimer's and dementia usually focuses on younger individuals. But by showing some of the research that we're doing in these two different cohorts of oldest old individuals, I hope that I can convince you that the oldest old are an important group to study. And it's an important group to study as their own and not necessarily assume that they are just older versions of older adults. So why the oldest old? So thanks to increases in life expectancy, decreases in birth rates, and the baby boomer generation, the population, the elderly population is growing very fast. And in particular, the oldest old, that are people 90 and older. They're the fastest growing segment of the population in the US and in many countries around the world. And from census data, we see that currently we have about 2.5 million people over the age of 90, but that number will more than triple by 2060 to about 8.5 million. And the oldest old are not growing only in terms of numbers, but also as a percentage of the older population, or those that are 65 and older. And we see here that it goes from about 4% of the elderly population to almost 10% of the elderly population. And that growth is also in many countries around the world, and the most dramatic of it is in terms of numbers is in China, where you can see that there's an estimated 31 million people 90 and older by 2060. And 90-year-olds have very high rates of comorbidities, physical disabilities, and have the highest rates of dementia, as you will see later, and many require a lot of care. So considering how the population and the age structure is changing, we are facing a huge public challenge, a huge public health challenge to care for the oldest old. And as an illustration of that, if we consider, for example, people 20 to 65 as the people available to care for those 90 and older, we can see how the ratio of caregivers to care receivers has been declining very rapidly. So while in 2001, there were about 99 potential caregivers for every care receiver, that ratio will go down quickly to about 22 to 1 by 2050. So again, creating a big challenge to the health system and how to care for these individuals. So even with these very large numbers and these staggering numbers, the oldest told have been systematically excluded from most research. They usually are excluded from national and international studies like ATME. They're excluded from most clinical trials. So there's really a lack of information about this very important and very fast growing group. And population-based studies, which are the ones that I usually work on, usually have low numbers of people at these very extreme ages. And that's usually understandable because the needs for studying people at these ages are kind of different. And it's hard to design studies that are adequate for a whole wide range of ages, and as I will describe a little bit. So to address some of these challenges in studying the oldest told and to fill some of the gaps in the knowledge, we established the 90+ studies a couple of decades ago. That study began, basically the 90+ study is a population-based longitudinal study of aging and dementia that had its basis on another study, the Leisure World Cohort study, that was a study in a retirement community here in Southern California in the early 80s. So at that time, there were questionnaires about health and lifestyle that were sent to all the residents of that retirement community. And the survivors from that study that were age 90 and older, starting on January 1st, 2003, were then invited to be part of the 90+ study. And we were able to enroll about 80% of the people that were eligible from that original cohort. So there was a lot of information about them from the early 80s and then later on as we recruited them into our 90+ study. Later on, as the Leisure World Cohort participants were kind of dwindling because they were a very specified fixed number, and given evolving research aims in the study, and including new technology like imaging for Alzheimer's biomarkers, so we began recruiting from outside the cohort from volunteers in the surrounding community. And we do evaluations frequently, short, and then we put a lot of flexibility in place and time. And the evaluations are done every 6 months, so we can capture rapid change and fast change in cognition and function that happen at this advanced age. We had to design our procedures to overcome sensory impairments, which are very prevalent at this age, and fatigue. And actually, fatigue was an issue that we did not necessarily anticipate when we began our study. We very quickly understood that we needed to shorten all our assessments. So we selected short versions of the neuropsychological test and short versions of basically all the assessments that we were doing. We also allow visits to be completed in more than one meeting if necessary. We also offer the visits either at the research office or at their homes if they prefer. And we provide transportation when needed to come to the research if necessary. So we tried to facilitate their participation as much as possible to maximize that participation, to maximize data collection, and to maintain the population-based aspect of the study. We also rely heavily on questionnaire and information from informants to really capture some of those subtle changes that may happen. And we also do genetic studies, brain imaging that was introduced basically halfway through the study, and then a large donation program that began from the very beginning. And this is an aspect of the study that I'm going to show you a little bit about later, where I think we've learned the most in this study. So since we started the study actually has received a lot of attention from the media, and several of participants actually were featured in 60 Minutes, actually twice, most recently about 4 years ago. So we were getting people interested in this very important segment of the population. So these are sort of the baseline characteristics of the people when they join. So as you can see, there's about 3 women for every man, which is typical of that age range. The group is relatively highly educated, with about 42% having a college degree or more. But you can also see that there's no racial diversity in this cohort. They're almost all, the cohort is almost all white. And it reflects the composition of the retirement community in the early 80s and in Orange County, of 90-year-olds in Orange County when we began our study in 2003. We actually have recruited, the average age at recruitment is about 93, but we actually even recruited people that were as old as 110 when they joined our study. At one point, we actually had the oldest man living in the U.S. that was as part of our study. And in terms of cognitive diagnosis, we actually kind of have about a third of the people when they join the study, about a third have normal cognition, about a third have impairment without meeting criteria for dementia, about a third dementia. And in terms of the etiology, we say that the majority of them are Alzheimer's disease, but a diagnosis, a clinical diagnosis or the etiological diagnosis at this age is kind of hard to really come to, and you'll understand later as I present some more information about why really trying to divide them into discrete groups is really not necessarily feasible at this age. For most of the study, the main outcome has been dementia. So one of the first things we did was to estimate the rates of dementia. And here there's incidence rates in studies that include people 90 and older. So the belief was that if a person reached age 90, a person was no longer at risk, or the risk would go down. And other studies actually here suggested that maybe the rates kind of either plateau or flatten or go down, although most kind of still kept going up. And that's kind of what we also saw in our study, that the rates continue to increase with age. They double approximately every 5 years and reach about 40% per year in centenarians. And we were one of the first groups who actually published incidence rates in centenarians. And with these very high dementia, and the rates were very similar in men and women, which is something that not necessarily everybody always agrees on. Very often people say that, well, the risk of dementia is higher in women than in men. That is not what we saw at all. We actually saw very similar rates in men and women. The differences are in the centenarians, where actually we have very few men at that time. So the estimates are a lot less precise, as you can see in the confidence interval. So with these very high rates of dementia, and coupled with the increase in the numbers in the 90+ population, we will have more cases of dementia among the oldest old in the coming decades that we currently have at all ages combined. So again, this is kind of setting up to be a huge public health challenge in the coming years in relation to, in terms of the amount of the vast number of people that we're going to have at this rate at this age with dementia. So the next steps in our studies were kind of to identify factors that were related to the presence or the absence of dementia. There were many factors, I mean, basically trying to understand what are the factors that increase the risk or lower the risk of developing dementia after age 90. We studied a variety of these factors that have been associated with dementia in younger elderly, like vitamin intake, BMI, alcohol intake, physical activities, leisure activities. And in reality, we found that, I mean, none of these were related to the risk of dementia in the oldest old. So not even an ApoE genotype, which is, we know that is a very strong genetic risk factor for Alzheimer's disease and dementia in younger elderly. So once people make it to this age, their ApoE4 genotype was not related to a risk of dementia. So basically, the risk factors for dementia are not necessarily the same in young versus oldest old. Some of the other things that we studied are vascular risk factors. So a lot of research suggesting that different types of cardiac and cerebrovascular conditions have increased the risk of dementia. And this is so far what we found. So in people who entered our study at age 90 and older without dementia, what we see is surprisingly vascular diseases, and some of the risk factors seem important. We see that congestive heart failure, heart valve disease, stroke are significantly associated with a higher risk of dementia. As you can see, the relative risk above 1 on that graph. For other conditions, although the estimates implies a higher risk. The evidence was not as clear. But interestingly, vascular risk factors like high cholesterol and hypertension are associated with a lower risk of developing dementia at this age. So we see that people in their 90s who have a history of hypertension have a lower risk of developing dementia. And this is contrary to what we know about these risk factors earlier in life, since research consistently shows that midlife hypertension and midlife high cholesterol are associated with a higher risk of dementia. So it seems like the association with dementia changes with age for some, but not all vascular diseases. Some diseases really do continue to have an increased risk, but others have a decreased risk at very advanced ages. So one thing to keep in mind is that what is high for these risk factors is usually determined by cut points that are established from studies with younger people. So what is an optimal blood pressure or an optimal cholesterol for a 60 or a 70-year-old may not necessarily be optimal for a 90-year-old in terms of preserving cognition. So we explored that result with hypertension a little bit further. And because we had information, remember that I said that the participants were originally part of this other leisure role study that had information collected earlier in life, about 15 to 30 years earlier. We combined the information about hypertension from that information that was collected in the leisure role study with information from later in life. And we were able to sort of to kind of estimate the age of onset of hypertension. And what we found was that what matters was when hypertension developed, not if. And here we see a graph that's comparing groups to people that do not have a history of hypertension in green. And we see that people who develop hypertension earlier in their life, like in their 70s or earlier, the risk is not different for people that do not have a history of hypertension. But those that develop hypertension in their 80s or more, even more in their 90s, they have a lower risk of dementia. So what our research suggests is that some of the factors that are related to dementia in the oldest adult are not necessarily the same as they may be in younger elderly. There's definitely several hypotheses for what this result may be. Our favorite one is that perhaps at this age, given the changes in the blood vessels people at that age throughout life, that maybe a higher blood pressure is necessary to maintain adequate perfusion to the brain. There's also other groups that actually believe that this is more of a selection bias and that in reality, we're just studying people that are very different and people that don't live to that age. But I guess our argument against that is that, well, if it's that people don't make it to this age, how come other factors like stroke and heart disease are still looking like they have an increased risk, but these other risk factors are not? So perhaps it's a combination of both, but we do really believe that perhaps really at this age, a higher blood pressure may be necessary to maintain adequate cognition. An important aspect of our studies also try to understand the sort of the neuropathologies and the underlying changes in the brain that are underlying cognitive impairment at this advanced age. And that's why we also have imaging of the autopsy sub-studies. So we're all investigating basically imaging biomarkers. We do amyloid PETs, tau PETs, and MRIs. Here, kind of showing basically the cross-sectional association of the imaging biomarkers in cognition, in relation to cognition. And we see that for amyloid, obtained from an amyloid PET, we clearly see that there's an increase, basically a clear association with cognition, where a higher amyloid deposition with increasing cognitive impairment. And basically the amyloid deposition is different between all the groups, and you can actually sort of distinguish between the groups. And we see sort of the same thing with hippocampal volume. Again, difference between the different cognitive diagnosis groups. One thing that I kind of also want to mention is that we do actually have a very large distribution with people with normal cognition that actually have very high levels. And so That's actually a part of the study that later on I'm going to talk a little bit about, but probably not going to get too much into it in terms of basically resilience, or people that are able to maintain high levels of cognition with very high levels of pathology in their brain too. So one thing that was interesting is that when we looked at white matter hyperintensities, which is sort of a measure potentially of cerebrovascular disease, we didn't actually see a difference in the for the different cognitive groups. But that was sort of a cross-sectional association that was sort of at the time of the scan. So what about progression? Are these biomarkers related to the rate of cognitive decline? This is work that was actually recently done by a PhD student doing a rotation with us, is that the answer actually is yes. So in this graph, I mean, for illustrative purposes, we have the low group. It's represented by the 25th quartile, and the high group in the 75th quartile. So you can see them here. And this kind of shows the trajectories for the mini-mental, like basically a test of global cognition. And we see the black line is the solid black line, is that people actually have low amyloid and high hippocampal volume. That is the group that declined the slowest. The group that have both high amyloid and low hippocampal volume declined the fastest. And what's interesting is that we actually see the difference is that the solid line and the dashed line is basically the same combination, but in people with and without white matter hyperintensities. And what we see is that amyloid accumulation and hippocampal volume or lower hippocampal volume are associated with a baseline cognition and the rate of cognitive decline. But white matter hyperintensities do not seem to be associated in this group. And I have to say that we were a bit surprised by the strong association with amyloid, because some research has suggested that the strength of the association with Alzheimer's disease neuropathologies weaken with advanced age. Here, we did not necessarily see that. We see that that's still very strongly related to cognition and cognitive change. But we also were very surprised by the white matter hyperintensities lack of association, because we know that cerebrovascular disease is very common at this age. So we were kind of surprised at the lack of the association with white matter hyperintensities, and we're not really clear and understand exactly why that may be. Maybe, I guess, after accounting for the other things, then white matter hyperintensities do not matter, or maybe what's being measured in the MRIs is not necessarily reflecting what's happening in their brain later on in terms of the actual neuropathology. So that to us was a very interesting finding. So we're learning a lot from imaging, but ideally we could see the potential reason for cognitive impairment during life, and imaging is kind of helping us do that in many cases, but there's still many of the diseases and brain abnormalities that cause dementia, especially at this age, that can only be seen by looking directly at the brain after death. And we actually have, like I mentioned, we have a large program with brain donation where we've already accumulated about 510 autopsies from our volunteers. And many of our major findings have been through this brain donation program. So we consider it a very, very important part of the research. We have identified many neuropathological changes associated with dementia at this age. We have Alzheimer's disease, Lewy body disease, hippocampal sclerosis, late or limbic predominant age-related TDP-43 encephalopathy. That is a newly appreciated and newly described condition that it mimics Alzheimer's disease. That is particularly relevant for the oldest old. We also have microvascular lesions that are strokes that are too small to be seen, and then conditions that are affecting the small blood vessels and the larger blood vessels, and then CAA, or accumulation of amyloid in the walls of the blood vessels. So most of these cannot be seen through brain imaging or detected during life at this time. And people with these pathological abnormalities are not usually recognized or are often misdiagnosed, usually with Alzheimer's disease. And again, only through postmortem examination of the brain is that we can really identify them. And most of the prevalence for these also increases with age and are most frequent in the oldest adult. So here we see the frequency and the severity of the changes in our study. We see basically the shading represents the severity of the neuropathology for each of the neuropathological abnormalities. We can see that Alzheimer's disease neuropathology is very common. Based on the Alzheimer's NIA, Alzheimer's Association criteria, we have more than 2/3 of our participants have intermediate or high levels of neuropathology. We also see that almost everybody have neurofibrillary tangles, and more than 90% have stages of 3 or higher. On the far side, we see that vascular neuropathologies are also very common, especially in the mild and moderate levels. And in the middle, we kind of have the other neurodegenerative changes, including late, that again, more recently described, that is about 1/3 of our participants. Lewy bodies are a lot less frequent in our group. So, so as I mentioned, I mean, many of these cannot be seen during life, but they're very common at this age. When we look at the contribution of all of them to dementia, we essentially see that all of them, most of them, actually are significantly associated with the odds of dementia, even after adjusting for each other. And they all kind of have similar odds. We see like Alzheimer's disease on the left here. We see that about people that die with that pathology in their brain are twice as likely to have dementia as people without. On the far right, we actually see atherosclerosis, severe atherosclerosis, and 3 or more microvascular lesions, and late, where with odds ratios about close to 3. So 3 times more likely to have dementia if they die with these neuropathologies. And if we add the neuropathologies all together, we see that compared with people that have none or one of these neuropathological changes, we see a very strong stepwise increase in the odds of dementia with increasing numbers, getting to a very, high odds ratio of 16 with people with 5 and more neuropathologies. And also notice that with the gray bars at the bottom, that almost 90% of the participants with dementia have 3 or more of these abnormalities. So these abnormalities are very common in our participants, and they actually appear together. So dementia is really due to multiple neuropathologies, and often many of them at the same time. And when we use that information about the prevalence of these and the magnitude of the association with dementia, we quantify the proportion of cases that could be averted if we eliminated the association. If we eliminated basically the different neuropathologies. So what proportion of cases could be attributed to each of these pathologies? And when we consider the individual pathologies here, we see that Alzheimer's disease in red and arteriolosclerosis in blue have the highest and very similar attributable fractions, about 26 and 24%. We have late, that is about 17%. So again, it's sort of the next one of the neurodegenerative conditions. Amyloid angiopathies in the 10. When we look at combinations of neuropathologies, we see that we combine sort of all the blue arteriolo, CAA, atherosclerosis, and microvascular lesions. We see that combined, they account for about 38% of all the cases. And the non-AD neurodegenerative, Lewy body, late, and HS account for about 28%. So we often say that Alzheimer's disease is the most common type of dementia, but that is not the case in the oldest adult. So you can see that Alzheimer's disease actually accounts for about 1/4 of the cases at this age. But given that most research is focused on eliminating amyloid, even if we completely eliminated Alzheimer's disease neuropathology, we would still have about 3/4 of the dementia cases at this age. So this means that interventions that are targeting a variety of diseases, not just Alzheimer's disease, are very needed to really significantly reduce the burden of dementia in this fast-growing segment of the population. And as I mentioned before, there's research that others have reported that the association of AD neuropathology and dementia weakens with age. And at least in our cohort, we didn't actually see that. This is work that was done by a medical student a couple of years ago, or published last year, where we looked at the, actually we have about, we published it with 100 centenarians. So when we actually got to 100 centenarians, we did this work on comparing the prevalence and the likelihood of dementia in nonagenarians and centenarians, centenarians in blue. And what we see is that we may not necessarily have some significant difference, but we clearly see no indication that the association with dementia attenuate. And in reality, the odds ratios for centenarians are higher than for nonagenarians for pretty much every single one of these pathologies. So again, it doesn't look like the association attenuates with age, as other people have suggested. And the multiple pathologies shown here basically at the higher gray and green in these bars, again, are more common in centenarians, and that stepwise association with the number of pathologies even more dramatic in centenarians, as you can see here. So increasing odds of dementia with increasing number of pathologies, again, are more so in centenarians than nonagenarians. So the strength of the association of the individual pathologies and in the number of pathologies, unfortunately, appears to get stronger with age, even at very advanced ages. So given the presence of these multiple pathologies and that many still cannot be identified during life, a big push in the field, and in particular, one of our collaborators, Dr. Ahmad Sajadi, is to kind of try to understand what people with those neuropathologies may look like clinically. So for example, do their cognitive profiles differ, in particular for late or the condition having to do with TDP 43? As you saw before, late is an important neuropathology, and especially in the oldest old, is about 30% prevalent in our cohort, and the prevalent increases with more severe impairment. And we're actively exploring the clinical presentation and other characteristics of people with different neuropathologies. Here in this table, we see like the association between neuropathologies and the different cognitive domains. And we see some similarities in the presentation, but we see some differences too. So for example, we see that people with memory deficits are likely to have both Alzheimer's disease neuropathology or late neuropathological changes, but they differ in other domains like visuospatial function that was appear in Alzheimer's disease and Lewy bodies, but did not appear in people with late. But then orientation, on the other hand, appear in people with late, but not, or was more likely in people with late, but not Alzheimer's disease. So by understanding the specific presentations or cognitive profiles, we hope to maybe eventually be able to identify the right people for the right clinical trial or the right treatment, or for the right treatment when available. What's interesting is how these also neuropathological changes kind of overlap often and co-occur in these individuals. This is sort of a histogram with all the different combinations of the neuropathological findings. These basically, each row represents each different neuropathology, and the columns represent the number of people with each of the different combinations, going from the right with no neuropathologies all the way to the left, people that have 7 neuropathologies. And I think what's and in the top we have the colors. No dementia is in red, and dementia is in blue. So you can see that towards the right there's more red, meaning no dementia. Towards the left there's blue, meaning more dementia. But we see that many people with multiple neuropathologies, like 4, 5, even 6 neuropathologies, we actually have people with that many neuropathologies that do not have dementia. And this kind of brings us to, I guess, the concept of resilience. I'm not going to spend much time on that concept here, even though it's very interesting for our group. But this brings me, like, this person, basically, we published as a case report a couple of years ago, a 96-year-old retired engineer that maintained normal cognition throughout life. You can see the neuropsychological scores here, the mini-mental, Boston naming, the memory test, the CBLT, perfect all the way to the end. Scores for all the neuropathologies were around the 90th percentile. This person had more comorbidities, TIAs, atrial fibrillation, congestive heart failure, developed hypertension at age 91. And this participant even had an ApoE4 allele, which we know is associated with an increase with dementia. This person also had bilateral lacunar infarcts on a CT scan. And when this person died, this person had Alzheimer's disease. You can see amyloid plaques, neurofibrillary tangles. This person had Lewy bodies. This person had late. So this person was essentially resilient to three or even four of the neuropathologies, and still was able to maintain normal cognition all the way throughout life. So this is a particular topic of particular interest, and normal cognition, basically in the presence of multiple dementia pathologies, is rare, as we saw from the graphic before, but it's still possible in the oldest old. And that's one of the active areas of research in our group right now. So now I'm switching gears a little bit, because everything that I've told you so far are results essentially in wide research participants. As I told you before, the 90+ study has very little racial and ethnic diversity. That reflected the composition of the area when we started the study. But that is not the case anymore. Here in the US is clearly becoming more diverse, and that is also true for the oldest old. By 2060, of the 8.5 million people that are 90 and older, about a third of them, or more than a third of them, will be of ethnicities other than white. And that is increasing from less than 5% when we began our study 20 years ago. And as far as we know, there's really only one study that's looking at dementia rates in diverse populations of the oldest old. In a study by Dr. Paula Gilsanz at Kaiser Permanente of Northern California, she used information from the Kaiser system, the electronic health record from the Kaiser system. And we can see that the racial and ethnic differences in rates of dementia that are seen in younger elderly seem to continue after age 90, with Black and Latino individuals having the highest rates of dementia. But there is really, up to that time, no information from cohort studies of diverse populations of the oldest old. So that's when we joined forces with colleagues from UC Davis, like Rachel Whitmer and Paola Gilsanz, like I said, from Kaiser Division of Research in Northern California, to start the study Life After 90. So this is an ongoing multiracial cohort of oldest old individuals. Participants have been longtime members of the Kaiser Permanente of Northern California integrated health system. They have to be proficient in English, either English or Spanish. And at the time of recruitment, they cannot have had a diagnosis of dementia in their medical record. We began enrollment in 2018, and we have so far enrolled 1,196 participants. And you can see on the right side, the real true ethnic and racial diversity of this cohort. We also have optional imaging and brain donation program that is it's sort of slowly building up. So I'm gonna present a few sort of preliminary studies from this. And one thing that's very exciting about this study, that this can actually be a true life course longitudinal study. We have, first, for all the participants, we had information from the Kaiser medical record system that goes back many years, because they started the system back in 1996. So we have a long period of time with information from their medical records. And then for many of them, we also have some additional information from midlife as part of this multiphasic health checkup that was started by Kaiser as a program to collect additional information from their members for research purposes. So again, we actually have information about midlife social economic status, workplace exposure, and a lot of midlife health indicators. So it's a very interesting and very rich data set that is gonna allow us to ask many interesting questions. As part of the study, we also ask questions about cognition, function, discrimination, social standing, social networks. So many interesting questions that we can explore. We started the study in 2018, in the next cycle that just began a couple of years ago, we are recruiting additional members and added a few additional measures. So we learned a lot from the 90+ study in ways of how to design a study for 90-year-olds. And we do evaluations every 6 months to, again, capture fast changes. We do short assessments, again, to avoid fatigue. And we do all the evaluations at home. Actually, we travel to their homes to facilitate participation. The evaluations are actually split in half. We actually have 2 teams, and some of the assessments are done by the Kaiser team, and some of the assessments are done by the DATAS team. Again, makes the evaluation shorter and makes it easier on the participants, again, to maximize participation at this age. So some of the characteristics of our participants, the average age is about 92. In terms of the sex distribution, we have about 62% of them are women. That proportion is a little bit lower in the Asian individuals, where it's closer to half. We definitely have a higher educational attainment among our Asian and white individuals, with more than half having a college education. That proportion is much lower in our Latino and Black participants, as you can see here. And this is sort of the average geriatric depression scale score is about 2.9, but Latinos clearly have a higher score, endorsing more of the items in the depression scale. This is a cool map that one of our students just prepared recently, showing the countries of birth of our participants, and we actually have 36 different countries of birth represented. After the US, we have China, the Philippines, Mexico, El Salvador, Japan are the most represented. So really one of a kind and truly multi-ethnic cohort of oldest old individuals. Here are some of our preliminary estimates of incidence. The rates seem to be a bit higher for women versus men. We have big differences by age, as we expected, with the rates more than doubling after 5 years. Also decreasing dementia rates with increasing education, that as seen in most cohort studies. And we now also see higher risk of dementia rates in our Black and Latino participants. That is, again, consistent with many studies, especially in younger populations. So if I superimpose the rates from the work that I presented earlier by Dr. Paula Gilsans from this same pool of people, with the estimate base that were estimated from the electronic medical records in green, we see that our rates are higher in general than what it was estimated from the medical records. So it seems like there's underdiagnosis of dementia at this age. Not surprisingly, I think it happens at many ages, at most ages, but it's also suggesting some important potential difference and even a larger underestimation in our Black and Latino individuals. So again, this is kind of supports the value of the research, where we are enrolling a cohort with a short 6-month follow-up. So it's important to really, again, with this age group being the fastest growing segment with the very high rates of dementia, it's very important to really understand the magnitude of the problem, as dementia seems to be underdiagnosed. And so the estimates from the medical records may be a real underestimate at this age group, and in particular in some specific ethnic groups. Most of the work that we've done so far has been in trying to understand what may be related to differences in cognitive abilities initially, what may be related to decline, and what may be related to those differences in decline. Here we have graphs showing cognitive domains scored by the CINAS battery, basically the Spanish English neuropsychological assessment scale that was actually developed by neuropsychologist Dan Mungus at UC Davis. And this is sort of also looking at the battery over a maximum of 4 years of follow-up. We see that we have significant difference for the different groups. For example, we here see that white participants performing much better at the time of entry, and the Latino participants performing worse. Also here in semantic memory, again, white individuals performing better. But the differences seem to only be at baseline. We actually do not see clear differences in the rates of decline. As you can see here, basically these are pretty straight slopes and parallel slopes. So no clear difference in the rates of decline. So we are, again, exploring what may be accounting for some of these differences at entry. And we see that education has a very strong association. We see people, the lines in red, basically the people with less or a high school education or less at entry would perform much worse, either whether it's a male, men or women, in executive function, semantic memory. These are sort of the more dramatic differences. Again, the large differences in baseline scores by educational attainment, but no differences in rate of decline. So now we have actually work that has been done by many different people that are kind of shown here in terms of different factors that are being looked at in relation to these. And here's sort of a summary of all that work. I kind of tried to arrange them here in some kind of temporal order from the factors that may have influences earlier in life to factors that influence later in life. And we see a very strong, a strong education effect at baseline for all domains indexed by the CNAS battery. But again, no association over time. We also see that everything so far that we have looked at is related to executive function. And what is interesting is that the factors that we think that may play a role earlier in life, like education, the country of birth, and time when they immigrated, are related to better baseline scores in semantic memory or acquired knowledge, while factors that play a role later in life seem to be related to better baseline scores for verbal memory or verbal episodic memory. And so far, we really have almost nothing related to cognitive change. So again, differences in the baseline scores and when people show up, but not over time. And perhaps we don't really have much in terms of follow-up because we only have an average of about 1.5 years so far in terms of follow-up. I mean, again, we've been sort of continuous enrolling. But could also be that people come in with a series of lifestyle experiences that in a way determine how well they perform initially when they join our study. But once we account for those differences, really there's no difference in the way or their progression over time. And this is important, but when we're trying to determine, for example, cut points for deciding impairment versus no impairment, or if a person meets a certain criteria for clinical trials, that perhaps due to the difference in their life course and experiences, people are not really starting at the same levels, and then maybe not necessarily we're comparing the same things, or the scores don't necessarily mean the same in different people. And we will continue to monitor this with additional follow-up, and we're actually exploring a variety of other factors that already work going on. We also have work on discrimination, midlife cardiovascular factors, early life SES. So exciting research going on. So like I said, we also have an imaging component. And these are kind of preliminary numbers, but here we see the age-adjusted means for amyloid accumulation seen by PET, white matter hyperintensities, and hippocampal volume. And we see, the numbers are still too low to really attempt to say anything definitive about difference in the groups. But what we found interesting is that there's some discordance on the burden of some of these biomarkers for some of the groups. Like, for example, we see that our Black individuals have the lowest average amyloid burden, but among the highest white matter hyperintensity volume. But conversely, our Latino individuals have the highest amyloid burden, but the lowest white matter hyperintensity volume. So maybe there's some initial suggestions that maybe the etiology of cognitive impairment may be different in the different groups. I think we're running out of time, so I'm kind of just go quickly to the last thing that I wanted to show. Again, we also have a brain donation program. We've already had about 116 of these volunteers that have come to autopsy. This is sort of a similar graph that I showed before in terms of neuropathologies. Again, Alzheimer's disease and vascular neuropathologies are the most frequent at this age. Again, levels of tangles. Almost every single person have some level of tangles. But what's interesting in this cohort so far is that the most severe levels, like of neurofibrillary tangles or even the high likelihood, is actually not common at all in this cohort. So that's kind of different from the other group. So it still remains to be, we still need to understand if there are real differences, methodological difference, but still a lot to learn about these pathologies and how they relate to cognition. And one thing I wanted to finish with is this something that I just learned about. This is our enrollment rate in our brain donation program back in 2021, when we had about 540 active participants. We actually see some differences in the enrollment rates for the different groups, much higher as you would expect with our white individuals, much lower in our Black individuals. But one thing that actually was very exciting to see that now in June of 2024, when we looked at the numbers, it was very exciting to see that the refusal rates are down, the enrollment rates are up, and it's nice to see that the numbers are very similar in all the different groups. So this speaks volume about the staff who perform the evaluations, that interact with the participants, and the relationships that they have built to actually make people comfortable actually joining these very important study. So this truly made my day the other day when I actually saw the numbers, and we think that we'll have an incredible, unique resource where we will be able to learn a lot from these amazing research volunteers. And so I guess to summarize, we've seen remarkable increases in longevity and in the number of oldest old, with increasing ethno-racial diversity of that group. The risk of dementia is exceptionally high, and in the coming decades, most of the cases of dementia will be in the oldest old. And the oldest old are not just older versions of younger elderly. The risk and protective factors are different in the oldest old versus the younger old. In the oldest old, dementia is the result of multiple neuropathologies, but most cases are not due to Alzheimer's disease. And my colleague, Dr. Sajjadi, likes to say that the RD and the ADRD is a lot more significant in the oldest old than at younger ages. And in the Life After 90, the multi-ethnic cohort, we saw that many factors seem to play a role across the life course are associated with cognition at baseline, but not necessarily with change. And there's an initial indication of the ethno-racial differences in the levels of neuropathologies that are consistent with those in younger ages, in younger elderly, but it's still too early to test for differences. Of course, the work is essential for advancing biomarker cognitive research at this age. And there's still a great need to continue investigating dementia and cognition in the oldest old, and especially in this diverse population who is severely underrepresented in research. So still a lot to learn about aging and cognition in the oldest old. So quickly acknowledge all the large group of people that actually make this work possible, both at UCI and the 90 Plus and the Life After 90, and in particular, the participants and the families that actually make this possible. And thank you.
Thank you so much for this wonderful talk. I just wanted to highlight that these are really, really interesting studies. And like when you were talking about like the recruitment of these populations, I was like, it's just so surprising that you're like managing to recruit so many participants. But I think that you did mention kind of like key aspects about like doing evaluations at home and significantly reducing the batteries to make sure that you're able to gather that data. So that was really interesting. And
Right.
I have a question before we go to the Q&A. So you mentioned kind of like the protective and risk factors that are totally different compared to like elder individuals, and that was like really shocking to hear. And you mentioned the high blood pressure and the high cholesterol for like the initial study that you mentioned. So I was wondering like if you guys are going to also take a look at that in the Life After 90 study, especially for like Black and Latino participants.
Right, absolutely. And then the nice thing is that we actually, because we also have the medical records from many years before, we actually can pinpoint even better when these things actually started occurring, what type of medications they were taking, just in case the effects actually have to do more with medication, which is that's what seems to be happening in our cohort, that for cholesterol, it seems to be maybe more about the medications. For blood pressure, it seems to be more about when the hypertension actually develops. But the other thing is that because we also have the larger Kaiser system and all the participants that made it to their 90s or did not make it to their 90s, we also can actually understand whether some of what we're seeing is actually has to do with that survival and selection biases. So that's why, I mean, this study is so amazing, because we not only have the participants that we enrolled, but we also have the whole history, all the medical records, even going back to midlife in terms of some of these factors, and the people that we didn't recruit, because we actually have a lot of information from the medical system, I mean, from the healthcare health records, to really understand what are the difference of the people that made it to the 90s, that enrolled in their study, and the people that did not. So it's a very rich data set that we definitely know that we're going to be able to learn a lot from.
Super exciting. I think that you guys are going to find like really, really cool findings. So we have a couple of questions here in the Q&A. So someone was asking, what do we know about social connectiveness and isolation in this population? Like, do you have any insight about that?
So we actually have somebody that was actually a postdoc and now is a faculty member that is actually looking at that right now, because we have been collecting a lot of information as connectivity. And I think I showed a little bit of that before, if I remember correctly, some of the, so we actually have some information about social networks. Basically, it was functional networks, and basically the number of people that are around them and how close they feel to other participants. And so far, I mean, that's still work that is kind of ongoing. But what we see again is that it seems to matter in terms of the levels of cognition when they enter. So people with better social networks are actually doing better cognitively. But that doesn't seem to be actually affecting the longitudinal course. But of course, we're actually going to be looking at that more in relation to a lot of other factors, not just cognition, but biomarkers and even pathology and a lot of other outcomes.
Great. So we have a couple of more questions if you still have time. So someone else was curious about, like, have you found any associations between like these brain resiliency with their BMI? So like perhaps like lower BMI across years show kind of like more active lifestyle, because exercise has been shown to increase brain health.
Right. In the 90+ study, we did not find anything having to do with BMI. What we do see, though, is that actually what was interesting, and I mean, I guess what the big people call the BMI paradox, or the obesity paradox, that from that Leisure World Cohort study, people that have at older ages, I mean, people in their 70s and 80s with a higher BMI actually were related to lower mortality, which again, sort of supporting the BMI paradox. We have not really seen anything related to cognition or even brain pathologies in relation to BMI. So, but now actually in the Life After 90, we actually have a lot more information that we can do, because we can actually go much further back in time, as all that information is actually available from the medical records. And for a large subset of them, it's also available from midlife. So we actually have a graduate student that is actually exploring a lot of these BMI, midlife BMI in relation to multiple outcomes in Life After 90.
Great, thank you. Someone else was also wondering, like, if you see if the brain resilient seniors have kind of like great social lives, meaning like living with their significant other rather than being single or widowed, versus like living in a community like a senior home, or like living in a city versus like a rural area.
Well, I mean, we haven't, in our 90+ study, we don't actually have most people in the rural areas. They all at one point lived in this retirement community in Southern California that is basically a suburb. One thing that we actually have seen in resilience, though, is that most of the participants at this age are women, and most of them are widowed women. And we do not actually find a difference by sex in terms of resilience. Actually, people that actually make it to this age with superior cognition, they're equally men and women. So far, I mean, we haven't actually looked at directly yet at their marital status, but the fact that most of the women that are in our study are widowed compared to the men, and we don't have a difference by sex, I think it's unlikely that we're actually going to have a difference by that. We haven't looked in terms of actually additional things in relation to maybe social networks, because we actually, unfortunately, we don't have that much information in the 90+ study about that. Again, we're hoping to do looking at some of that in our Life After 90.
Then someone else was also asking if you have found any impact of the medication statin use and the incidence of dementia among these groups, like in these age groups.
Right. So the result that I showed before about history of high cholesterol being related to a lower risk of dementia, that seems to actually be driven by statin use. So in reality, it seems to be like the medications themselves, the statin medications, actually seem to be what's actually related to a lower risk, which is different from what we found in hypertension. That it seems like the medications actually do not seem to have an effect at all, and it's really when the hypertension developed. But with high cholesterol, it actually seems to be the medications, the statins, that are actually driving that association.
Great. So I think those were all the questions that we had on the Q&A. I just wanted to thank you again for this wonderful presentation. Really, really interesting.
So yeah, thank you for having me, and thank you for listening.